200,000 years ago, there were so few of us that geneticists call it a bottleneck. Possibly just a few thousand humans on the entire planet, fewer than the student body of an average American university today. Before that, hundreds of thousands of years of living in caves and under rock overhangs while Neanderthals and Denisovans occupied Europe and Asia. And further back still, 7 million years of trial and error, extinct species, and dead-end branches. Human evolution was not a march of progress from ape to us. It was a story of near total failure. Several times we nearly disappeared entirely, and each time something, climate, accident, fire, language, gave us one more chance. This is the story of those chances. Before we go any further, please subscribe to this channel. Your support really helps us to continue this work. Thank you. There is a question that sounds simple, but contains within it the entire complexity of what we are about to explore. How did a creature with a brain the size of an orange, living in the retreating forests of Eastern Africa 7 million years ago, become the species that built the pyramids, landed on the moon, and is watching this video right now? The answer is not a straight line. It is not a ladder with rungs labeled in order. It is a bush, tangled, branching, with most branches ending in extinction and only one, improbably, reaching the present. And the most important thing to understand before we begin is that the branch that survived was not obviously the best one. It was not the strongest, not the largest, not even, for most of its history, the most numerous. It was simply the last one standing. Act 1. The world before us. The world in which this story begins bears little resemblance to the world we know. 7 million years ago, the continent of Africa was in the middle of one of the most consequential geological transformations of the past 20 million years. The East African Rift System, the enormous network of geological faults that runs from the Red Sea south through Ethiopia, Kenya, Tanzania, and into Mozambique, was actively tearing the eastern edge of the African continent apart. This rifting process was not merely geological scenery. It was the engine of human evolution. As the rift widened and the eastern highlands rose, the climate of eastern Africa changed fundamentally. The warm, moist air from the Indian Ocean that had previously watered the forests of eastern Africa was increasingly blocked by the rising rift escarpments. The forests began to fragment. The grasslands, the savannas, expanded into the spaces between the shrinking forest patches. And in those spaces, something new was forced to happen. The forests of the late Miocene, before the rift transformed them, were the home of the ape radiation, one of the most diverse assemblages of large-bodied primates the world has ever seen. Dozens of ape species inhabited the forests of Africa and Eurasia, far more diversity than the 8 living great ape species that survive today. These Miocene apes were forest specialists. They lived in the trees, ate fruit, and moved through the canopy with the swinging, grasping locomotion that their anatomy had been optimized for over millions of years. Most of them went extinct as the forests shrank. A few survived. And among the survivors, in the forests of what is now Central Africa and the woodland margins of what is now Eastern Africa, were the ancestral populations from which all living great apes, including humans, would eventually descend. The split between the lineage leading to chimpanzees and the lineage leading to humans is one of the most studied and most debated events in all of paleoanthropology. The molecular clock, the technique that uses the rate of accumulated genetic mutations to estimate when two lineages diverged from a common ancestor, places the human-chimpanzee split at somewhere between 7 and 5 million years ago. The uncertainty in that range reflects genuine biological complexity. Speciation is not an instantaneous event, and the human-chimpanzee split appears to have involved a prolonged period of population contact and possible hybridization before the two lineages became fully reproductively isolated. The last common ancestor of humans and chimpanzees was not a chimpanzee. It was not a human. It was something we have never found in the fossil record, in complete enough form to describe fully. A creature that would have looked broadly ape-like by modern standards, but that carried within its genome the potential for both of the lineages that descended from it. The oldest candidate for a member of the human lineage, a fossil that may represent one of the first organisms on the hominin side of the human-chimpanzee split, is Sahelanthropus chadensis, known from a single partial skull and some teeth found in Chad in 2001 and dated to approximately 6 to 7 million years ago. The skull, nicknamed Tumai, is one of the most debated specimens in paleoanthropology. It has a small brain case, roughly 360 cubic centimeters in volume, comparable to a modern chimpanzee, and far below the roughly 1,400 cubic centimeters of a modern human. Its face, however, is flatter than a chimpanzee's, and the position of the foramen magnum, the opening at the base of the skull through which the spinal cord passes, is positioned in a way that suggests the head may have been balanced on a vertical rather than a horizontal spine. A vertical spine means an upright posture. An upright posture means bipedal locomotion, walking on two legs. If Sahelanthropus was indeed walking upright, this is one of the most important facts in the entire human story. Because bipedalism, the defining anatomical characteristic of the hominin lineage, appears to have evolved at the very beginning of our split from the chimpanzee lineage, millions of years before any significant increase in brain size. For decades, the popular understanding of human evolution assumed that large brains came first, and everything else followed. The fossil record says the opposite. The body came first. The brain came much, much later. And this reversal of the expected sequence raises a question that paleoanthropologists have been grappling with for half a century. Why did we stand up before we got smart? The most compelling answer connects bipedalism directly to the ecological transformation that the East African rift system was producing. As the forests fragmented and the grasslands expanded, the distances between forest patches increased. An animal that could move efficiently across open ground from one forest patch to another had a significant advantage over one that could not. Bipedal locomotion, walking on two legs, is not faster than quadrupedal locomotion for a primate body plan. But it is substantially more energetically efficient over long distances. A biped uses significantly less calories per kilometer traveled than a quadruped of comparable body mass. In a world where food was becoming less dense and more widely dispersed, the energy saved by bipedal locomotion was the difference between survival and starvation over the course of a season. There was a second advantage, easily overlooked. Bipedalism freed the hands. And free hands, in an animal already capable of sophisticated object manipulation from millions of years of arboreal life, were a platform for a cascade of subsequent innovations that would eventually transform the world. The hands that were freed by standing up were the hands that would eventually make tools, control fire, draw on cave walls, and type on keyboards. Bipedalism was not merely a locomotor innovation. It was the enabling condition for everything that followed. Approximately 6 million years ago, in what is now Kenya, lived another candidate early hominin, Orrorin tugenensis. Known from a fragmentary collection of bones including femur fragments, arm bones, and teeth, Orrorin is significant primarily for what its femur reveals about locomotion. The femur head, the ball that fits into the hip socket, shows the distribution of cortical bone that is characteristic of bipedal loading. An animal that walks upright puts weight on its femur differently than an animal that moves quadrupedally, and the bone responds by thickening in characteristic locations. Orrorin's femur shows this bipedal loading pattern. 6 million years ago, in the East African woodland, something was walking upright. These earliest members of the human lineage were not impressive animals by any conventional measure of biological success. They were small, probably not much larger than modern chimpanzees, with body masses of perhaps 30 to 50 kilograms, about 66 to 110 pounds. Their brains were not significantly larger than those of living apes. They lived in woodland environments that were themselves under pressure from the changing climate, eating a diet that probably included fruit, leaves, seeds, and whatever animal protein they could obtain through opportunistic foraging. They had none of the behavioral or technological innovations that we associate with humanity. They were, by any objective measure, marginal animals in a world that was being transformed around them. But they were walking upright, and that, at this stage in the story, was everything. Act 2. The Australopithecines, a body without a mind. 4.4 million years ago, in what is now the Middle Awash region of Ethiopia, lived a creature that represents the most complete picture we have of the earliest stages of human evolution. Ardipithecus ramidus, known from a remarkable partial skeleton nicknamed Ardi, described in a series of landmark papers in 2009, was a hominin that defies easy categorization. It was bipedal, but its bipedalism was different from ours. It walked upright on the ground, but also retained grasping feet adapted for moving through trees, suggesting it spent significant time in both environments. Its canine teeth were smaller than a chimpanzee's, suggesting reduced male-male combat, and possibly the beginnings of the pair-bonding social structure that characterizes modern humans. Its brain was approximately 300 cubic centimeters, no larger than a chimpanzee's. Ardi tells us something crucial about the nature of the hominin lineage at this stage. It was anatomically transitional in virtually every respect. The feet that could grasp branches had not yet committed fully to terrestrial bipedalism. The teeth that showed reduced canines had not yet evolved the complex differentiated dental formula of later hominins. The brain that was no larger than a chimpanzee's had not yet begun the expansion that would eventually produce human cognition. Ardipithecus was in every sense a creature in between, not the ape ancestor and not yet the more recognizably human australopithecines that followed it. It was the experiment before the experiment. The australopithecines, the group of hominins that dominated the African fossil record from roughly 4 to 2 million years ago, represent the most diverse and, in many ways, the most confusing chapter in human evolution. They were bipedal, but their brains remained small, averaging between 400 and 500 cubic centimeters across most of the group. They had bodies that showed a mosaic of ape-like and human-like features, upright posture and bipedal legs, but long arms, curved fingers, and a skeletal anatomy that still bore the marks of a recent arboreal ancestry. They lived in a variety of environments across eastern and southern Africa, from the woodland margins of the East African Rift to the more open grasslands of the Turkana Basin, to the cave-dotted limestone landscapes of South Africa's cradle of humankind. The most famous australopithecine, and arguably the most famous fossil in the history of paleoanthropology, is Australopithecus afarensis, the species to which the skeleton known as Lucy belongs. Discovered in November 1974 by Donald Johansen and Tom Gray in the Afar region of Ethiopia, Lucy is a partial skeleton dated to approximately 3.2 million years ago. She was small, approximately 1.1 meters, about 3 feet 7 inches, tall and roughly 29 kilograms, about 64 pounds, and her anatomy shows the characteristic Australopithecine combination of bipedal legs and more ape-like upper body. Her knee joint and hip anatomy are unambiguously those of a biped. She walked upright. Her skull is small, her face prognathic, her brain approximately 430 cubic centimeters. She was a bipedal ape, not a primitive human in any meaningful biological sense. But Lucy's world was not just Lucy. The Letoli footprints of Tanzania, a trail of bipedal footprints preserved in volcanic ash dated to 3.6 million years ago, show that at least 2 and possibly 3 individuals of Australopithecus afarensis walked across freshly fallen volcanic ash at a site in what is now northern Tanzania. The footprints are remarkably human-like in their overall form, showing a bipedal gait with a rounded heel strike, a weight transfer pattern across the midfoot, and a toe-off from the big toe that is characteristic of human walking rather than the more lateral footplant of great apes. The Laetoli footprints are the oldest direct evidence of bipedal locomotion in the hominin fossil record, and they show that by 3.6 million years ago, Australopithecines were walking with a gait that a modern observer would immediately recognize as human-like. The Laetoli footprints also show something poignant and specific: the tracks of 2 individuals walking side by side, and within the tracks of one of them, the overlapping prints of a smaller individual walking in the footsteps of the larger, as if a child were playing the game of stepping exactly in a parent's footsteps. This is, of course, an interpretation that may overread the fossil evidence. But the image it conjures of individuals walking together across a landscape that was about to be buried in volcanic ash is one of the most humanizing images in the entire fossil record. 3.6 million years ago, our ancestors were social animals moving through the world together. And then, in August of 2025, everything we thought we knew about this period of human evolution became significantly more complicated. A team led by Brian Villmois of the University of Nevada, Las Vegas, and Kay Reed of Arizona State University, working at the Lady Giraru research site in the Afar region of Ethiopia, published a paper in the journal Nature that fundamentally revised the picture of human evolution at its most critical juncture. The study reported 13 fossilized teeth from sediments dated between 2.6 and 2.78 million years ago. Most remarkable was not their age, though their age was itself significant, but what they represented. Some of the teeth belonged to the genus Homo, confirming the presence of the earliest known members of our genus at 2.78 million years ago. But another set of teeth, found in the same location at 2.63 million years ago, belonged to a completely new and previously unknown species of Australopithecus, one that was distinct from Australopithecus afarensis, distinct from Australopithecus garhi, and distinct from any previously described hominin species. The implications of this finding are profound. At 2.6 to 2.8 million years ago, the critical window when the genus Homo first appears in the fossil record, the hominin landscape of Eastern Africa was not the simple succession implied by the traditional march of progress model. It was a crowd. At minimum, early Homo and this new Australopithecus species were sharing the same landscape simultaneously. And in a May 2026 paper, a University of Chicago team reported a 2.6 million year old Paranthropus jaw from the same broader Afar region. Combined with the previously known Australopithecus garhi from the same time interval, the fossil record now suggests that as many as 4 distinct hominin lineages were living in Eastern Africa simultaneously at the moment when the genus Homo first appeared. 4 experiments with the idea of being human, running simultaneously in the same landscape, competing for the same resources, eating similar foods, walking on the same ground. Only one of those 4 experiments would survive to the present. The others, Paranthropus, Australopithecus garhi, the new Lady Giraru Australopithecus, would all go extinct within the next million years, leaving no descendants. But at the moment of Homo's origin, they were all there, all alive, all apparently viable. Kay Reed, one of the co-directors of the Lady Giraru research project since 2002, described the finding with characteristic precision. Human evolution is not linear, she said. It is a bushy tree. The image that most people carry in their minds of an ape gradually straightening up and becoming more human in a smooth progression is simply not what the fossil record shows. What the fossil record shows is a profusion of experiments, most of which failed. and one of which only barely, only improbably, succeeded. Paranthropus, the robust australopithecines, deserve particular attention as the most dramatic example of an evolutionary experiment that came very close to working. Paranthropus was characterized by an extreme adaptation for processing hard, tough plant foods. Massive molars, enormous jaw muscles, and in some species a pronounced sagittal crest on the top of the skull that served as an anchor for the jaw muscles. These animals were not heading in the direction of intelligence. They were heading in the direction of powerful, highly specialized feeding machinery. For approximately 1.5 million years, from roughly 2.5 to 1 million years ago, Paranthropus species coexisted in Africa with the early members of the genus Homo. They were not obviously less successful than their Homo contemporaries for most of this time. They were numerous, widespread, and well adapted to the foods available in their environments. And then they went extinct. Why did Paranthropus go extinct while Homo survived? This is one of the most actively debated questions in paleoanthropology, and there is no fully satisfying answer. The most likely explanation involves dietary flexibility. The extreme dental and jaw specializations of Paranthropus made these animals very good at eating certain foods, hard seeds, tubers, roots, but potentially less capable of flexibly exploiting the range of food sources that a more generalist dentition allows. When the African climate became more variable and less predictable in the late Pliocene and Pleistocene, with more pronounced and more frequent oscillations between wet and dry periods, driven by the orbital cycles of the Earth, the dietary generalists may have had a significant advantage over the dietary specialists. The ability to eat almost anything, rather than being optimally adapted to eat one thing, may be the core of what kept the Homo lineage alive when the climate turned. Act 3. The birth of Homo. Tools, fire, and the first exodus. 2.8 million years ago, in the Afar region of Ethiopia, a jawbone was lying in the dirt that would eventually be found, millions of years later, and named LD350-1. Published in 2015 by Brian Villemois and colleagues, this jaw fragment, dated to 2.78 million years ago, represented the oldest known member of the genus Homo at the time of its description. Its teeth and jaw anatomy showed a clear departure from the australopithecine grade. The premolars were smaller than those of Australopithecus, the jaw itself was narrower and less robust, and the overall morphology was consistent with a dietary shift toward higher quality foods that required less grinding and more cutting. LD350-1 was not Homo sapiens. It was not even Homo erectus. But it was unambiguously Homo. The genus had begun. The species Homo habilis, the handy human named for its association with stone tools, appears in the fossil record approximately 2.4 to 2.3 million years ago and persists until approximately 1.4 million years ago. Homo habilis had a brain approximately 600 to 750 cubic centimeters in volume, significantly larger than the Australopithecine average, and the first unambiguous evidence in the fossil record of the brain expansion that would eventually produce modern human cognition. Its face was more orthognathic, less projecting than the australopithecines, and its overall cranial anatomy began to approach the distinctively globular shape that characterizes the genus Homo. But it retained small stature, long arms relative to legs, and a general body plan that still bore the marks of its australopithecine ancestry. The Oldowan tool industry, named for Olduvai Gorge in Tanzania, though Oldowan tools have since been found at sites considerably older than the original Olduvai finds, represents the first unambiguous evidence of stone tool manufacture in the archaeological record. Oldowan tools are not elaborate. They consist primarily of flakes struck from cobbles or pebbles by direct percussion, hitting one rock against another with sufficient force to detach a sharp-edged flake that can be used for cutting. The flaked core itself, once a few flakes have been removed, can also function as a chopper. Making an Oldowan tool does not require extensive training or planning. Experiments show that naive human subjects can produce functional Oldowan-grade flakes within a few hours of instruction. But the production of these tools requires something that no non-human animal consistently demonstrates: the understanding that a rock can be deliberately modified to create a sharp edge, and the motor control to execute that modification with precision. The oldest known stone tools in the fossil record are the Lomekwian tools from West Turkana in Kenya, dated to approximately 3.3 million years ago, predating the earliest Homo by nearly 500,000 years. This discovery, published in 2015, demonstrated that stone tool production was not unique to the genus Homo and may have been practiced by late australopithecines. The Oldowan tools associated with Homo habilis and early Homo represent a refinement of this older tradition, rather than its invention from scratch. But the association of more sophisticated tool production with the larger brains of early Homo is not coincidental. The cognitive requirements for systematic tool production, planning, manual dexterity, understanding of rock mechanics, selected for precisely the neural expansions that characterize the early Homo brain. What did these tools allow? The answer is not primarily fighting or hunting, as popular imagination tends to assume. The primary use of early stone tools, documented through microscopic analysis of use-wear patterns on Oldowan flakes, was processing food, specifically cutting meat and cracking bones to access marrow. Bone marrow is one of the most calorie-dense and nutrient-rich foods available to a terrestrial animal. It contains high concentrations of fat, protein, and essential fatty acids that are critical for the energy-expensive process of brain growth and maintenance. The brain is the most metabolically expensive organ in the body. It constitutes roughly 2% of body mass, but consumes approximately 20% of the body's total resting energy budget. Feeding a bigger brain requires better food, and access to meat and bone marrow, provided by stone tools capable of cutting through hide and cracking bone, provided the caloric substrate for the brain expansion that the fossil record shows in early Homo. This is one of the most important feedback loops in the entire story of human evolution. Slightly larger brains produced slightly better tools. Slightly better tools provided access to higher quality foods. Higher quality foods supported the growth of slightly larger brains. And so the cycle continued, each turn of the wheel producing a brain a little larger and tools a little more sophisticated over hundreds of thousands of years and dozens of generations. The most consequential species in the genus Homo, the species that dominated the hominin fossil record for nearly 2 million years, and that represents the first undeniable evidence of a human-like body form, is Homo erectus. Appearing in the fossil record approximately 1.9 million years ago, Homo erectus was a different kind of animal from its Homo habilis predecessors. It was tall, up to 1.8 meters, about 6 feet in some populations, with long legs adapted for efficient endurance walking over large distances. Its gut was smaller relative to body mass than the australopithecines and early Homo, reflecting a higher quality diet that required less digestive processing. Its brain, averaging approximately 900 cubic centimeters across the range of known specimens, was roughly double the australopithecine average. And its post-cranial skeleton, the skeleton below the skull, was, for the first time in the hominin lineage, recognizably modern in its overall proportions. In December of 2025, a research team from Midwestern University, led by Scott Simpson, published a detailed reconstruction of a Homo erectus cranium known as DAN5 from the Gona site in Ethiopia, dated to approximately 1.5 million years ago. The reconstruction, published in Nature Communications, produced a surprising result. The facial anatomy of this Homo erectus specimen was more primitive, more australopithecine-like than previous reconstructions of the species had suggested. Rather than the emergence of Homo erectus, representing a clean, sudden transition to a fully modern-style facial anatomy, the DAN5 reconstruction suggested that the transition was more gradual and more complex, with some populations of Homo erectus retaining facial features that previous researchers had assumed were confined to earlier hominin grades. The story of Homo erectus, like the story of human evolution generally, was messier than the textbooks had implied. The control of fire is one of the most debated topics in paleoanthropology because the evidence for it is archaeologically elusive. Fire leaves traces: burned bone, charcoal, heat-altered sediment that can persist in the archaeological record. But distinguishing deliberately controlled fire from natural brush fires is not always straightforward. The earliest convincing evidence for controlled fire use by hominins comes from Wonderwork Cave in South Africa, where burned bone and plant material dated to approximately 1 million years ago has been interpreted as evidence of fire use by Homo erectus. Evidence from Gesher Benot Yaakov in Israel, dated to approximately 790,000 years ago, shows burned wood and seeds associated with stone tools in a context that strongly implies deliberate fire control rather than accidental burning. Richard Wrangham of Harvard University has argued in his cooking hypothesis that the control of fire and the regular cooking of food was the most transformative event in the evolution of the human body, more important, in his view, than the evolution of stone tools. Cooked food is more easily digestible than raw food, releasing more calories and nutrients from the same quantity of food. Cooking also detoxifies many plant foods that are inedible raw. The energetic consequences of cooking are substantial. Cooking dramatically increases the net caloric value of both plant and animal foods, effectively expanding the caloric budget available for the expensive process of brain growth and maintenance. Wrangham argues that the reduction of the gut in Homo erectus, visible in the narrower pelvis and chest of this species compared to earlier hominins, reflects the evolutionary response to cooking, as the energetic savings from shorter gut tissue were redirected to brain growth. Whether or not cooking was established as early as the anatomical changes in Homo erectus would require, the control of fire also had enormous non-nutritional consequences. Fire extended the productive hours of the day beyond sunset, enabling the social gathering and communication that cumulative culture requires. Fire at the cave entrance excluded large predators. Fire provided warmth in cold environments. Fire was the first technology that fundamentally altered the relationship between the hominin lineage and the physical environment. Homo erectus was also the first member of the human lineage to leave Africa. The evidence for this is found in the spectacular fossil site of Dmanisi in the Republic of Georgia, where multiple Homo erectus skulls dated to approximately 1.8 million years ago were discovered in the 1980s and 1990s. The Dmanisi hominins were small, smaller than most African Homo erectus specimens, with brain sizes ranging from approximately 600 to 700 cubic centimeters, and they were associated with Oldowan-grade stone tools rather than the more sophisticated Acheulean hand axes that characterize later Homo erectus in Africa. The Dmanisi discovery demonstrated that the first members of the human lineage to leave Africa did so very early, within a few 100,000 years of Homo erectus's first appearance in the African record, and that they did not require either large brains or sophisticated tools to accomplish this dispersal. The expansion of Homo erectus out of Africa and into Eurasia over the following 1.5 million years produced a pattern of geographic differentiation that researchers are still working to understand. In Eastern Asia, the Homo erectus populations that arrived in China and Java evolved in relative isolation from the African populations, producing regional variants, sometimes called Homo ergaster for the African form and retaining Homo erectus for the Asian forms, that show distinct anatomical characteristics. The Sangiran and Trinil specimens from Java and the Jukodian specimens from Beijing, formerly known as Peking Man, represent the most extensively studied examples of Asian Homo erectus, and their anatomy shows both the core characteristics of the species: large brow ridges, elongated low skull, thick skull walls, and regional peculiarities that reflect hundreds of thousands of years of separate evolution. Axt 4, 1 million years of dominance and the quiet splitting. 1.5 million years ago, the stone tool industry underwent its first major transformation since the invention of Oldowan tools. The Acheulean industry, named for Saint-Acheule in France, where characteristic tools were first described, though the industry originated in Africa, introduced the hand axe. a large, bifacially flaked stone tool with a carefully shaped edge running around its entire perimeter, produced through a manufacturing process that required significantly more planning, skill, and cognitive foresight than the simpler Oldowan tools. A hand axe is not made by simply striking flakes from a cobble. It is made by envisioning the final three-dimensional form in the raw material before any flaking begins, then systematically removing material from both faces to produce the desired shape. This requires the ability to hold a mental template of the desired end product and to execute a sequence of precisely controlled actions to achieve it. Cognitive scientists refer to this as hierarchical planning, the ability to organize behavior into nested sub-goals that collectively achieve a larger goal. It is a capacity that appears to be uniquely well developed in the human lineage. The Acheulean hand axe is also remarkable for its extraordinary longevity. The industry appears in the African record approximately 1.7 million years ago and persists, with remarkably little change, until approximately 300,000 years ago. 1.4 million years of essentially the same tool design made by dozens of different hominin populations across Africa and Eurasia. This technological stasis is one of the most puzzling features of the Paleolithic record. The human brain was getting larger during this period. The average brain volume of Homo heidelbergensis, the late Acheulean toolmaker, was roughly 1,200 cubic centimeters, approaching the modern human average. Why did brain expansion not produce more rapid technological change? The most likely answer involves the nature of culture in the absence of cumulative cultural transmission, the ability to teach and learn innovations that build on previous innovations. Without language capable of conveying complex technical instructions, each generation was largely limited to learning by observation, and innovations that were not immediately and visually obvious were difficult to transmit. Homo heidelbergensis, the species that inhabited Africa and Europe from approximately 700,000 to 200,000 years ago, represents the common ancestor of both Neanderthals and modern humans. The species is defined primarily by a combination of characters: a brain approaching or equaling the modern human range in size, a skull that is more rounded than Homo erectus, but retains robust brow ridges and other archaic features, and a body that was robust and heavily built. Homo heidelbergensis populations in Africa were ancestral to Homo sapiens. Populations in Europe were ancestral to Homo neanderthalensis. And somewhere in Central Asia, a third lineage was diverging that would eventually leave us the most enigmatic record in all of human evolutionary history. In February of 2026, a team from the Max Planck Institute for Evolutionary Anthropology published a paper describing hominin fossils from the Thomas Quarry 1 site in Morocco, specifically from the Grotta a Hominides, a cave system whose sediments had remained undisturbed, providing what the researchers described as an undisputable stratigraphic context. The fossils were dated to approximately 773,000 years ago, making them among the oldest hominin fossils from northwestern Africa, and their anatomy suggested they may represent the shared common ancestor of both Neanderthals and modern humans, a population that was ancestral to both lineages before the split that eventually produced two separate species. If confirmed, The Thomas Quarry fossils provide a face, or at least a skull, for the common ancestor that geneticists have long inferred from the molecular data. The divergence of the Neanderthal and modern human lineages from their common ancestor is dated by the molecular clock to approximately 700,000 to 500,000 years ago. This range aligns reasonably well with the anatomical evidence. Early Neanderthal features appear in the European fossil record beginning approximately 400,000 years ago, and the lineage leading to modern humans begins showing distinctively modern features in Africa, beginning approximately 300,000 years ago. The two lineages diverged from a common ancestral population, evolved in geographic separation for hundreds of thousands of years, and eventually met again, with consequences that are still visible in the genomes of every non-African human alive today. Act 5. A crowded world. The branches we did not expect 60,000 years ago. The world was not the lonely place it appears in the human imagination when we project our current solitude backward in time. It was occupied by multiple different kinds of humans. This is perhaps the most dramatic revision in our understanding of human evolution over the past 2 decades. The discovery that our species, Homo sapiens, shared the planet with at least 5 other species of humans within the past 100,000 years. Not distant evolutionary predecessors, not fossil curiosities, but contemporaneous populations of humans who were, in some cases, our neighbors, our competitors, and, as ancient DNA has now definitively shown, our mates. The Neanderthals are the best known of our extinct relatives, and the story of Neanderthal intelligence has undergone one of the most dramatic revisions in the history of paleoanthropology. For most of the 20th century, Neanderthals were depicted as brutish, dim-witted cave dwellers, the stereotype that gave the word Neanderthal its colloquial meaning as an insult. The fossil and archaeological evidence has systematically demolished this characterization over the past 30 years. Neanderthals had brains that were, on average, slightly larger than modern human brains, approximately 1,400 to 1,500 cubic centimeters. They made hafted spear points using a multi-step manufacturing process that required planning and technical skill comparable to modern human tool-making. They buried their dead, sometimes with possible grave goods, a behavior that implies a concept of death and possibly of an afterlife. They used pigments, ochre and manganese dioxide, to decorate their bodies. They may have made personal ornaments from eagle talons and shells. And they cared for their injured and ill. The Shanidar cave skeleton from Iraq shows an individual who had suffered severe injuries, a withered arm, a damaged eye, and survived for years, implying that others in the group provided food and care. The Denisovans are the most mysterious of our extinct relatives, and the story of their discovery is one of the most remarkable in the history of science. In 2010, a team led by Svante Pääbo at the Max Planck Institute for Evolutionary Anthropology published the analysis of DNA extracted from a finger bone found in Denisova Cave in the Altai Mountains of Siberia. The DNA was neither modern human nor Neanderthal. It came from a previously unknown human population that had interbred with both Neanderthals and modern humans. The Denisovans, named for Denisova Cave, were known, for years afterward, only from a finger bone, a few teeth, and a jaw fragment found in Tibet. They had no face. They were ghost humans, present in the genetic record of living people but invisible in the fossil record. In 2025, that changed. Analysis of DNA extracted from the dragon man skull, a large, archaic-looking human skull found in China in 1933 and kept hidden for decades before being donated to a university in 2018, revealed that Dragon Man belonged to the Denisovan lineage. As CNN science journalist Ashley Strickland reported in December 2025, the analysis electrified the scientific community, providing the first cranial anatomy that could be confidently attributed to the Denisovans. The skull is large and robust, with a massive brain case and archaic facial features that place it clearly outside the range of modern human variation. It is, by any conventional measure, a different kind of human, and it was alive in China perhaps as recently as 146,000 years ago. The Denisovan genetic legacy is visible today in the living populations of the Asia-Pacific region. Modern Melanesians and Aboriginal Australians carry approximately 4 to 6% Denisovan DNA, the highest archaic admixture of any living human population. Tibetans carry a specific Denisovan gene variant, the EPAS1 gene, that allows them to live at high altitude without the red blood cell overproduction that causes altitude sickness in other populations. This gene was not evolved independently by Tibetans. It was acquired through interbreeding with Denisovans, who had already adapted to high-altitude environments in Central Asia. The Denisovans shaped the biology of their descendants in ways that are still saving lives today in Tibetan villages at 4,000 meters, about 13,000 feet, elevation. Homo floresiensis, the Flores hominin, nicknamed the Hobbit for its diminutive stature, was discovered in 2003 in Liang Bua Cave on the Indonesian island of Flores, and dated to approximately 100,000 to 60,000 years ago. Adults were approximately 1 meter, about 3 feet 3 inches, tall, with brains of approximately 400 cubic centimeters, roughly the size of a chimpanzee brain. Their small body and brain size is most likely explained by island dwarfism, the well-documented phenomenon in which populations isolated on islands tend to evolve towards smaller body sizes over time due to the limited food resources available in island environments. Homo floresiensis was apparently making stone tools of a sophistication comparable to contemporaneous modern human tools, an extraordinary feat for a brain of such limited volume, and a finding that challenges assumptions about the relationship between brain size and cognitive capability. Homo luzonensis, discovered in 2019 from fossils found in Calayo Cave on the Philippine island of Luzon and dated to approximately 67,000 years ago, extends the geographic range of small-bodied island hominins further into the Pacific. Like Homo floresiensis, Homo luzonensis shows a mosaic of primitive and derived features that has made it difficult to place confidently within the known hominin phylogeny. Its discovery demonstrated that the island chain of Southeast Asia hosted not one, but multiple independent experiments in small-bodied hominin evolution, each developing in isolation on islands separated by the deep water channels of the Pacific. Homo naledi, discovered in the Rising Star Cave System in South Africa's Cradle of Humankind in 2013 and described in 2015, is perhaps the most anatomically enigmatic of the recent hominin discoveries. Its brain was small, approximately 460 to 610 cubic centimeters, comparable to early Homo. Its overall anatomy showed a confusing mixture of primitive and derived features, with feet and legs that were remarkably modern in their proportions, and hands and shoulders that were more australopithecine. Its geological age, when finally determined in 2017, proved to be shockingly recent, between approximately 335 and 236,000 years ago. Homo naledi, with its small brain and primitive upper body, was alive at the same time as early Homo sapiens in Africa, and the cave context in which its remains were found, accessible only through extremely narrow passages in a location far from any natural entrance, has been interpreted by some researchers as suggesting deliberate mortuary behavior. If Homo naledi was intentionally depositing its dead deep in a cave system, this represents complex symbolic behavior in an organism with a brain no larger than a chimpanzee's, a finding that fundamentally challenges the assumption that symbolic behavior requires modern human brain size. Act 6. Homo sapiens, the cognitive explosion and the exit from shadow. 300,000 years ago, in the Jebel Irhoud region of Morocco, lived a group of hominins whose fossils, described in 2017 by Jean-Jacques Hublin and colleagues, represent the oldest known remains of anatomically modern Homo sapiens. The Jebel Irhoud fossils, multiple individuals including adults and juveniles, show a brain size and overall cranial capacity in the modern human range, but with a brain case that is more elongated from front to back than the distinctively globular skull of modern humans. This elongation suggests that the posterior parietal and temporal regions of the brain, regions associated with complex spatial cognition, social cognition, and language, were not yet fully expanded to their modern form. The Jebel Irhoud hominins had modern human faces and modern human brain sizes, but not yet fully modern human brain shapes. They were, in the terminology that paleoanthropologists use, anatomically modern, but not yet behaviorally modern. What happened to the brain in the interval between 300,000 years ago and the emergence of fully modern human behavior approximately 70 to 100,000 years ago is one of the most actively studied questions in cognitive neuroscience and paleoanthropology. The transition to behavioral modernity, the suite of behavioral characteristics that define modern human cognition, including symbolic art, complex language, long-distance trade, and the cumulative cultural transmission of innovations, does not appear in the archaeological record as a sudden event. It appears gradually, with different elements appearing at different times and in different places across Africa. The Blombos cave site on the southern Cape Coast of South Africa is perhaps the most important single archaeological site for understanding the emergence of behavioral modernity. Excavations at Blombos, led by Christopher Henshelwood of the University of Bergen and the University of the Witwatersrand, have produced a series of finds dated to between 100,000 and 70,000 years ago that document symbolic behavior of striking sophistication. Ochre pieces engraved with geometric patterns dated to approximately 77,000 years ago represent the oldest known intentional geometric designs made by humans. Shell beads, perforated for stringing as personal ornaments, dated to approximately 75,000 years ago, are among the oldest known personal ornaments in the archaeological record. A toolkit for producing red ochre pigment, including grinding stones, mixing bowls made from abalone shells, and the ochre itself, dated to approximately 100,000 years ago, represents the oldest known evidence of systematic pigment production and possibly paint making. The ochre engravings of Blombos are not merely interesting artifacts. They are evidence of a cognitive capacity that distinguishes modern humans from all other animals. The ability to create and communicate meaning through abstract symbols. A geometric engraving on a piece of ochre conveys no survival-relevant information to anyone who does not share the cultural context that gives the pattern its meaning. Making such a pattern requires the ability to hold an abstract concept in mind, a symbol whose significance is culturally assigned rather than naturally obvious, and to translate that concept into a physical form that can be preserved and transmitted. This is, in a profound sense, the cognitive signature of modern humanity. It is the beginning of a process that eventually produces writing, mathematics, science, and the kind of long-form documentary you are watching right now. The genetic bottleneck is one of the most sobering facts about our species. Genetic analyses of the diversity of the human genome, using multiple independent methods, including mitochondrial DNA, Y chromosome analysis, and genome-wide single nucleotide polymorphism data, consistently indicate that at some point in the history of our species, the population of Homo sapiens was dramatically reduced, possibly to somewhere between 1,000 and 10,000 breeding individuals. The timing of this bottleneck is estimated at approximately 150,000 to 70,000 years ago, with some analyses pointing to an event approximately 74,000 years ago that corresponds to the eruption of the Toba supervolcano in Sumatra, the largest volcanic eruption in the past 2 million years, which may have produced a volcanic winter severe enough to dramatically reduce food availability across Africa and Eurasia. Whether the Toba eruption was the direct cause of the genetic bottleneck remains debated. The archaeological record from Africa does not show a clear gap in occupation sites that would be expected if the continent's human population had been nearly exterminated. But the genetic signal of an extreme population reduction in our evolutionary history is unambiguous. At some point in the past 200,000 years, our species came close to extinction. The entire genetic diversity of the approximately 8 billion humans alive today descends from a population that may have been smaller than the current student enrollment at a single large university. The exit of Homo sapiens from Africa, the second great exodus of the human lineage following the earlier exodus of Homo erectus, is documented by both the fossil and genetic records. The most widely accepted model places the primary dispersal event at approximately 60 to 70,000 years ago, when a population of modern humans left northeastern Africa, possibly through the Sinai Peninsula or across the southern end of the Red Sea at the Bab el-Mandeb Strait, and began the expansion into Eurasia that would eventually carry the species to every habitable corner of the planet. The genetic evidence for this expansion is found in every living non-African human. The Neanderthal DNA that makes up 1 to 4% of the genomes of all non-African living humans was acquired during the period when expanding Homo sapiens populations first encountered Neanderthal populations in the Middle East and Europe. The genomic evidence, analyzed by David Reich of Harvard and Svante Pääbo at the Max Planck Institute for Evolutionary Anthropology and their respective teams, indicates that this interbreeding occurred approximately 50 to 60,000 years ago, consistent with the archaeological evidence for the presence of modern humans in the Levant and Western Asia at this time. The Denisovan DNA that is present at elevated levels in populations from South and Southeast Asia, and especially in Melanesia and Aboriginal Australians, reflects a separate interbreeding event, or possibly multiple events, between expanding Homo sapiens populations and Denisovan populations in Asia. Recent genomic analyses suggest that the ancestors of Melanesians and Aboriginal Australians may have encountered and interbred with two genetically distinct Denisovan populations, one more closely related to the Siberian Denisovans known from Denisova Cave, and one more distantly related that may represent a separate population of Denisovans that had been isolated in Southeast Asia for hundreds of thousands of years. The extent of the interbreeding between Homo sapiens and archaic humans was, it appears, considerably greater and more geographically complex than was apparent from the first wave of ancient DNA analyses. The implications of this for how we think about ourselves are profound. Modern humans are not the product of a pure lineage descending unbroken from a single African population. We are a hybrid species, carrying genetic contributions from at least 3 and possibly more archaic human populations. The Neanderthal genes in European and Asian genomes include variants associated with immune function, skin and hair characteristics, and neurological development. The Denisovan EPAS1 gene in Tibetan populations is saving lives at high altitude right now. We did not replace the archaic humans. We absorbed them. They became part of us, and we carry them forward in our DNA and in our biology, even as we were the ones who ultimately survived. Act 7. The Conquest of the World, from Africa to every shore. 65,000 years ago, the coastline of what is now the Indonesian archipelago looked very different from today. Sea levels were approximately 120 meters, about 394 feet, lower than they are today, because enormous quantities of water were locked up in the glacial ice sheets of the last glacial maximum that was then approaching its peak. Much of what is now the shallow Sunda Shelf, the continental shelf between the Malay Peninsula and the islands of Borneo, Java, and Sumatra was dry land, forming a vast extension of the Asian continent called Sundaland. The islands that lay beyond this exposed land, New Guinea, the Bismarck Archipelago, the Solomon Islands chain, were not connected by land to Sundaland. Between Sundaland and what is now New Guinea lay deep water channels that required water crossings even during the glacial low stand. Sometime between approximately 65 and 50,000 years ago, a group of modern humans crossed those water channels. The genetic evidence for this crossing is found in the ancestry of all Aboriginal Australians and New Guineans, populations whose ancestors represent the first humans to colonize the Australia-New Guinea landmass, called Sahul, during the glacial low stand when the two landmasses were connected by the exposed Sahul shelf. This crossing was not accidental drift on a log. It required deliberate watercraft capable of making open water crossings of at least 60 to 80 kilometers, about 37 to 50 miles, the minimum gap between islands even at glacial low stand. The colonization of Sahul represents the first unambiguous evidence of human seafaring, and it pushes the origin of deliberate watercraft navigation back to at least 65,000 years ago, tens of thousands of years before the earliest known boats in the archaeological record. The humans who arrived in Sahul encountered a world of extraordinary ecological richness and biological naivety. The Australian megafauna, the giant marsupials and reptiles that had evolved in isolation from placental mammals for tens of millions of years, had never experienced human predators. Diprotodon optatum, a wombat relative the size of a hippopotamus. Procoptodon goliath, a giant short-faced kangaroo standing approximately 2 meters, about 6.5 feet tall. Thylacoleo carnifex, the marsupial lion with the most powerful bite force relative to body size of any known mammal. These animals had no evolved fear response to upright bipedal primates carrying spears. They were, in the terminology that ecologists use, naive prey. Prey animals that had not co-evolved with the specific predation strategies that modern humans employ. The timing of the megafaunal extinctions in Australia correlates closely with the timing of human arrival. Most of the large Australian megafauna went extinct between approximately 50 and 45,000 years ago, within a geologically brief interval of the first documented human presence on the continent. The debate over whether these extinctions were primarily caused by human hunting, by the climate change associated with the approach of the last glacial maximum, or by some combination of both, has been ongoing for decades and is not fully resolved. The most compelling current evidence supports the synergy hypothesis. Climate change reduced population sizes and habitat connectivity, making the megafauna vulnerable, and human hunting delivered the killing blow to populations that were already stressed. The natural experiment of Wrangel Island, where a dwarf mammoth population survived until approximately 4,000 years ago, when humans first arrived on the island and went extinct within a few centuries of that arrival, provides the most direct evidence that human presence, rather than climate alone, was the terminal factor in many megafaunal extinctions. The colonization of the Americas presents a different set of archaeological puzzles. The genetic evidence is unambiguous. The founding population of the Americas was a single group that crossed from northeastern Asia into what is now Alaska via the Beringia land bridge, the exposed continental shelf between Siberia and Alaska that existed during the glacial lowstand. The timing of this crossing and the route by which the founding population then spread south into the Americas has been debated intensively for decades. The traditional model placed the colonization at approximately 13,000 years ago, based on the earliest well-documented archaeological sites in the continental United States, the Clovis sites, characterized by the distinctive Clovis projectile point technology. But a growing body of evidence suggests that the Americas were colonized earlier, possibly much earlier, by populations that reached South America before the Clovis horizon, and whose archaeological signature is less well preserved in the North American record. The Monteverde site in Chile, dated to approximately 14,500 years ago, 1,500 years before the oldest Clovis sites in North America, provided the first widely accepted evidence for pre-Clovis occupation of the Americas. The Pedra Furada site in Brazil and the Chiquihuite cave site in Mexico have produced dates that, if confirmed, would push the human presence in the Americas back to 60,000 years ago or more, though these claims remain contested. The most recent synthesis of the genetic and archaeological evidence suggests that the Americas were first colonized by people who crossed Beringia and followed the Pacific coastline southward approximately 16,000 years ago, possibly in watercraft rather than entirely on foot, reaching South America before they had fully colonized the interior of North America. In North America, the arriving humans encountered a megafaunal community as naively trusting as the Australian megafauna had been. Woolly mammoths, mastodons, Columbian mammoths, ground sloths, giant short-faced bears, dire wolves, American horses, and American camels. As in Australia, the correlation between human arrival and megafaunal extinction is striking. Approximately 35 genera of large North American mammals went extinct between approximately 15 and 10,000 years ago, a rate and selectivity that climate change alone cannot fully explain. The American horse, which had evolved in North America over the previous 55 million years, and whose descendants in the Old World would eventually be domesticated to power the agricultural civilizations of Eurasia, went extinct in its homeland within a few thousand years of the first humans crossing Beringia. The last glacial maximum, the period from approximately 26,000 to 19,000 years ago, when the ice sheets of North America and Eurasia reached their greatest extent, temporarily interrupted the human expansion into the high latitudes of both hemispheres. Ice sheets 3 kilometers, about 2 miles thick, covered what is now New York City. The tundra extended to the latitude of modern Washington, D.C. Sea levels were approximately 120 meters below today's, exposing vast areas of continental shelf as dry land and creating the Beringia land bridge between Asia and the Americas. Human populations in Europe and Asia retreated to southern refugia during the glacial maximum, areas south of the ice sheets where the climate remained sufficiently mild to support human occupation. These refugia, the Iberian Peninsula, the Italian and Balkan peninsulas, the Levant, Central Asia, Southeastern Asia, preserved the genetic diversity of the populations that had been dispersing across Eurasia for the previous 40,000 years. The patterns of genetic diversity in modern European populations record the bottlenecks and expansions of these glacial refugia populations with remarkable precision. By 20,000 years ago, Homo sapiens was the only surviving member of the genus Homo on Earth. The Neanderthals had gone extinct approximately 40,000 years ago, their populations declining over the 20,000 years following the arrival of modern humans in Europe. The Denisovans had disappeared, their last known populations in Siberia probably going extinct by approximately 40,000 years ago. Homo floresiensis on the island of Flores had gone extinct approximately 50,000 years ago, possibly following the arrival of modern humans in the Indonesian archipelago. The cause of the Neanderthal extinction remains debated. Competition with modern humans for resources, disease transmission from the expanding modern human populations, and climate change associated with the Heinrich events of the late Pleistocene have all been proposed as contributing factors. Most researchers today favor a synergy hypothesis similar to the one proposed for the megafaunal extinctions. Modern human population expansion reduced Neanderthal territory and resources, making them vulnerable to the demographic consequences of climate oscillations that they might otherwise have survived. The result was that, for the first time since the earliest members of the genus Homo appeared in the fossil record approximately 2.8 million years ago, there was only one kind of human on Earth. We were alone. And while this solitude is so complete and so enduring that it has become the baseline of human experience, the default assumption that has shaped philosophy, religion, and the arts throughout the brief history of human civilization. It is, on the scale of hominin evolutionary history, a remarkable and anomalous condition. For most of the past 2 million years, multiple kinds of humans existed simultaneously. Our current solitude is the exception, not the rule. Act 8. The Holocene and Civilization, from Hunters to Builders. Approximately 11,700 years ago, the climate of the planet crossed a threshold. The last glacial maximum was over. The ice sheets that had covered northern North America and northern Eurasia were melting. Sea levels were rising. They would eventually rise by approximately 120 meters over the following several thousand years, flooding the Beringia land bridge, submerging the continental shelves that had connected islands and continents, and creating the coastline geography we know today. Temperatures were warming rapidly, approaching and eventually exceeding the modern baseline. The Holocene, the geological epoch that we still inhabit, had begun. The Holocene is, by the standards of Pleistocene climate variability, an anomalously stable warm period. The dramatic glacial-interglacial oscillations that had characterized the past 2 million years, temperature swings of 10 to 15 degrees Celsius over cycles of 100,000 and 41,000 years, were replaced by a period of relative climatic stability in which the global average temperature varied by only 2 to 3 degrees Celsius over more than 11,000 years. This stability was not coincidental with the development of agriculture. It was its prerequisite. The agricultural revolution, the transition from hunter-gatherer lifestyles to settled farming communities that began independently in multiple locations around the world, between approximately 12,000 and 7,000 years ago was made possible by the climatic stability of the Holocene. Before the Holocene, the variability of the climate made it impossible to invest in the long-term cultivation of plant crops and the husbandry of domesticated animals that agriculture requires. A drought or a cold snap that killed the crops could eliminate the food supply of an agricultural community in a way that it could not eliminate the food supply of mobile hunter-gatherers who could move to find food elsewhere. The relative climatic stability of the Holocene reduced this risk to levels where the investment in agriculture was sustainable over generations. The Fertile Crescent, the arc of productive land stretching from the Jordan Valley through the Levant and into Mesopotamia and the upper Tigris-Euphrates Valley, was the site of the earliest and most extensively documented agricultural transition. By approximately 11,500 years ago, the Natufian culture of the Levant was intensively harvesting wild cereals, emmer wheat, einkorn wheat, and wild barley from the rich stands of wild grain that grew in the winter rainfall belt of the eastern Mediterranean coast. By approximately 10,500 years ago, communities at sites like Jericho in the Jordan Valley and Ain Ghazal in Jordan were cultivating domesticated emmer wheat and einkorn wheat, raising domesticated goats and sheep, and living in permanent or semi-permanent settlements. The agricultural transition in the Fertile Crescent was not a sudden invention, but a gradual process of intensification. A long period of pre-adaptation in which the Natufian hunter-gatherers became increasingly dependent on wild cereals and increasingly oriented toward the management and eventually the cultivation of those cereals. The domestication of crops involved a genetic transformation of the wild plant species that is visible in the archaeological record. Wild emmer wheat has a fragile rachis, the attachment between the seed and the plant stem, that shatters when the plant is ripe, dispersing the seeds to the ground. This is adaptive for the wild plant, as it ensures seed dispersal. But it is a serious problem for human harvesters who lose much of the grain before they can collect it. Domesticated emmer wheat has a tough rachis that retains the grain on the plant until it is deliberately harvested. This non-shattering rachis phenotype arose through genetic mutations and was selected by humans, consciously or unconsciously, over the generations of cultivation that followed the initial exploitation of wild cereals. The selection from tough rachis mutants among the wild population over hundreds of years of harvesting eventually produced a fully domesticated cereal crop whose seeds were entirely dependent on human harvesting and planting for their reproduction. Agriculture developed independently in at least 6 and possibly more locations around the world. The Fertile Crescent for wheat and barley, China for rice and millet, New Guinea for taro and yams, Mesoamerica for maize and squash and beans, the eastern United States for goosefoot and sunflower and marsh elder, and the Andes for potato and quinoa. The near simultaneity of these independent agricultural transitions, all occurring within the first 5,000 years of the Holocene, reflects the enabling role of the post-glacial climate rather than cultural diffusion from a single source. The stable warm conditions of the Holocene allowed independent agricultural development wherever suitable wild plant species were available, and population pressure created the incentive to intensify food production. The consequences of agriculture for human population were immediate and dramatic. Hunter-gatherer populations are constrained in their density by the carrying capacity of the local environment. The number of people that can be supported by the wild foods available in a given area. Agricultural communities, by producing food rather than merely harvesting it, can support much higher population densities than hunter-gatherers in equivalent environments. The transition to agriculture was accompanied everywhere by a significant increase in population density and, in many regions, a rapid expansion of farming populations into territories previously occupied by hunter-gatherers. The demographic consequences of agriculture were also, paradoxically, negative in important respects. The skeletal record of the agricultural transition shows a consistent pattern of health decline compared to the preceding hunter-gatherer populations. Shorter average stature, reflecting reduced nutritional diversity as diet narrowed to a few staple crops. Increased frequency of dental caries and enamel hypoplasia, reflecting the high carbohydrate diet of cereal farming. Higher rates of infectious disease, reflecting the crowding of dense permanent settlements and the close proximity to domesticated animals that are reservoirs for zoonotic pathogens. The agricultural revolution made the human population more numerous and more sedentary, but in many respects less healthy and less nutritionally diverse than the hunter-gatherer lifestyle it replaced. The first cities appeared in Mesopotamia approximately 5,500 years ago. The site of Uruk in modern Iraq, the city from which the biblical Erech was derived and whose name gives us the modern name Iraq, grew from a modest agricultural settlement to a city of perhaps 40,000 people by approximately 3,000 years BCE, making it the largest human settlement that had ever existed up to that point. The growth of Uruk was made possible by the agricultural surplus produced by the intensive irrigation agriculture of the Tigris-Euphrates floodplain, perhaps the most productive agricultural land on Earth in the ancient world, and by the social and political innovations that allowed that surplus to be collected, stored, redistributed, and used to support populations of specialized non-food producing craftspeople, administrators, priests, and soldiers. Writing appeared in Mesopotamia approximately 5,200 years ago, initially as a system of clay tablets for recording economic transactions, tallies of grain and livestock that were needed by the administrators of the large temple complexes that dominated the economic life of the early Mesopotamian cities. The earliest writing was not narrative. It was accounting. The first written records are inventories, receipts, and administrative lists. Literature, the use of writing to record stories, ideas, and the inner life of individual humans, came later, beginning with the Epic of Gilgamesh from approximately 4,000 years ago. But the transition from oral to written transmission of knowledge was one of the most consequential in all of human cognitive history. Writing allowed knowledge to be stored outside the human brain in physical media that could outlast any individual human life, be transmitted across distances without personal contact, and be accumulated across generations in a way that pure oral tradition cannot sustain. Writing is the technology that made the cumulative growth of human knowledge, science, history, philosophy, law, possible. The period from the first cities to the present, approximately 5,500 years, encompasses the entirety of recorded human history. Every civilization, every empire, every scientific discovery, every work of art and literature and music that we know about falls within this interval. It is the most intensively documented period in the 4 million year history of the hominin lineage, and it represents approximately 0.1% of the total duration of that history. From the earliest Australopithecus to the present is approximately 4 million years. The entirety of recorded human history is 5,500 years. The proportion of the total story that we can read in written records is roughly 1 part in 700. The other 699 parts are written in stone and bone and DNA, the other archives of the human past. Today, Homo sapiens numbers approximately 8 billion individuals. We occupy every continent, every climate zone from tropical rainforest to polar desert, every elevation from sea level to the permanent settlements at 4,000 meters, about 13,000 feet, in the Andes and the Tibetan Plateau. Our species has modified approximately 40% of the Earth's land surface for agriculture and has increased the concentration of atmospheric CO2 by approximately 50% above the pre-industrial baseline, a change whose geological consequences will be visible in the rock record millions of years from now. We have driven hundreds of species to extinction and are in the process of driving thousands more in what ecologists describe as the 6th mass extinction, the first caused primarily by the actions of a single biological species. We are, by any measure of ecological impact, the most consequential species in the history of complex animal life on Earth. More consequential than the dinosaurs, which dominated the land for 165 million years, but altered the planet's chemistry relatively little. More consequential than the cyanobacteria, whose photosynthetic revolution transformed the atmosphere 2.5 billion years ago, though the cyanobacteria did it over hundreds of millions of years, while we have accomplished our transformation in a few centuries. Now let me tell you what I think about all of this. As someone who has spent time with the fossil record and the genetic data and the archaeological evidence that tells the story of 7 million years of hominin evolution, the most important lesson of human evolutionary history is one that the popular story almost always gets wrong. We were not inevitable. At every point in the 7 million year story we have just traced, there were moments when the hominin lineage could have gone extinct, when the climate turned, when the food ran out, when the competition from other species or other hominins was too intense. The 4-species hominin crowd of Lady Geraru 2.8 million years ago included our direct ancestors as one among several experiments. The genetic bottleneck of 150,000 years ago reduced us to perhaps a few thousand individuals. The arrival of modern humans in Eurasia 60,000 years ago brought us into competition with Neanderthals, who had successfully occupied Europe for 300,000 years. At none of these points was our survival certain. At several of them, it was close to improbable. What saved us each time? Not intelligence alone. The Neanderthals had brains as large as ours and made tools as sophisticated as ours. Not strength alone. Homo erectus was larger and more robustly built. Not longevity. Homo erectus survived for nearly 2 million years compared to our 300,000. What saved us appears to have been a combination of things that, in retrospect, look less like advantages and more like accidents of circumstance. The particular ecological flexibility of our ancestors at Giraru, who could eat a wider range of foods than the Paranthropus specialists beside them. The particular social structure and information-sharing capacity that allowed us to cooperate across larger groups than Neanderthals. The particular climatic timing of our second exodus, which the Thomas Quarry research now helps us anchor more precisely in the record. And the most recent discoveries, the 2025 Nature paper from Giraru confirming the 4-species hominin crowd, the 2026 Thomas Quarry fossils from Morocco, revealing the face of our common ancestor with Neanderthals, the dragon man DN, identifying the Denisovans at last. These discoveries collectively tell a single consistent story. Human evolution was messier, more branching, and more improbable than any of the tidy march of progress diagrams that filled the textbooks of the 20th century ever suggested. The second lesson of human evolutionary history is that we are not as alone as we feel. Every non-African human carries 1 to 4% Neanderthal DNA. The Denisovans, whose face we finally saw in 2025, are present in us in ways that are still saving lives at altitude and shaping immune responses in the Pacific. The archaic humans we replaced are not entirely gone. They live in us, in our immune systems, in our metabolism, in the genetic variants that give specific populations specific adaptive advantages in specific environments. We did not succeed the archaic humans. We absorbed them, and they shaped what we became. The third lesson, the hardest one, is that our success is very recent and very fragile. 7 million years to go from a small-brained ape in a fragmenting forest to a species of 8 billion that is currently conducting an uncontrolled geological scale experiment on the planetary systems that produced it. The geological record shows us what happens when those planetary systems are disrupted beyond the tolerance range of the biosphere. The 5 previous mass extinctions in the fossil record are a library of consequences. We have not yet read the book. We have only written the first pages of the new chapter. Whether the genus Homo, the last member of which is us, will prove as durable as Homo erectus's 2 million year tenure suggests is possible, or as vulnerable as the population bottleneck of 150,000 years ago suggests, is also quite possible, is a question that the geological record cannot yet answer. It simply records. And what it records, in 7 million years of fossil bone and genetic sequence, and stone tools and charcoal and ochre engravings, is the story of a creature that was almost nothing, that nearly disappeared multiple times, that arrived at its current extraordinary capability by accident and adaptation and the narrowest of margins. We are here because of luck as much as intelligence, because of climate as much as culture, because of the particular accidents of geology and evolution that shaped us. And the full story of those accidents, from the first upright step in the fragmenting forests of the East African Rift to the first city on the Euphrates floodplain, is the most extraordinary story that has ever been told, because it is the only story that is ultimately about all of us.